morning, everybody. Just like my colleagues before me, I as well am very grateful to be here and very excited to be talking to you about my research over the past 5-ish years. As you can see, I slightly altered the title, and the reason for that is I really wanted to emphasize even further kind of the approach that we have in our research, which is essentially studying biological questions, but kind of building the systems from the ground up. And I'll talk to you about that. Okay, so what I'm really interested in biologically is understanding early embryonic development. And this is really the most exciting time of our lives, because in the first couple weeks of development after fertilization, we dramatically change shape, the protein expression landscape changes also dramatically. Cells become specified into tissues, we constantly break symmetries, and this is really a complex phenomenon that can be, and I kind of boil it down into 3 events. One of them being, it starts with signaling, then interpretation of these signals that forms patterns, and finally, we need to break symmetries in order to form the body plan as we see it. I have to tell you, as you heard, I'm not a biologist by training. Probably I'm best described as a chemical physicist, molecular physicist. And as many physicists doing biology, I really like to read, and this is not, I'm not just saying that, I really do like to read Darcy Thompson's On Growth and Form, which was published precisely 100 years ago, and I recommend you to look at it. This is this big book up there, so he was a mathematician who was just looking around in his environment, looking at all the life around him and trying to really understand the generalities, what makes life. And so he would use mathematics, physics, engineering to explain how the mechanics of walking or how the skeleton is formed. You can see here, he drew up these ganglion cells from various different animals, and he saw, well, they're not very different in size, but these animals are vastly different in size, somehow leading him to conclude, perhaps a little romantically, that life itself, or the complexity of life, is much more than the sum of its parts when comparing, say, humans and mice. Fast forward 100 years later, we still have a lot of physics in biology, and which really is interesting to me is that we still sometimes use some of these basic Darcy Thompson-like principles, and I highlighted an experiment here. And I highlighted in a paper from last year, which looked at early embryonic development in the mouse, and they found that interfacial tension between cells of the embryo is really driving the sorting of these cells. And as you can see, they use a mathematical description of interfacial tension exactly as it was used 100 years ago. But more prominently, physicists, what they do in biology today is try to understand dynamical systems. So we're trying to understand patterning. We're also trying to understand the vast complexity of gene regulatory network that really drives development. And for this, you need a quantitative approach. Another quote I like to show usually, and this is really boiling down what we are doing, and that means that what this quote really means is that as physicists, we find biology to be very complicated, right? So we want to find the most basic elements in biology that can be used to reconstruct a system. And if we are able to make it happen just as we see it in a cell, then we believe that we really understand something about that system. And this is sort of the leitmotif of my work as well. And so I'll show you 2 projects today that are sort of very different in a way, but also they have commonalities. And on the one side is, the first question is how cells receive these signals, and this is a process called cell trafficking and endocytosis, and it happens on a subcellular scale. And the other process is then how cells perceive this signal, interpret the signals to form a pattern. And so the common theme in both is forming these synthetic systems. So on the left side, when I studied trafficking, I would create synthetic cell membranes, whereas on the right side, what I'm doing more recently is I'm creating synthetic human embryos. Okay, so first question. The first step in cellular trafficking is called endocytosis, and it's a process by which cells receive nutrients, signals, pathogens, and basically, as you can see, this is the canonical picture of endocytosis in which a cargo enters the cells, invaginates the membrane, form this sort of vesicle, spherical membrane, which is coated by clathrin, and at the end of this process, you have a GTP-driven clipping of the membrane, which then releases the cargo into the cell's lumen. And this is a textbook case of endocytosis. However, our collaborators in Cambridge and us have found, and you'll see a movie here, have found that there is actually another process of endocytosis that is used by many signaling proteins and also pathogens that completely bypass this pathway and only require very few ingredients. So this pathway requires a lot of ingredients, and our pathway requires only a few ingredients. And you can see here that there are these worms that are entering into the cell. These are basically membrane tubes. So we're not even forming these vesicles. We're not using clathrin. We're just forming long tubes into the membrane, and the reason why you only see it briefly is because indeed it happens really fast. And the main components that we found were responsible for this process of entering the cells was curvature proteins, so the membrane proteins that specifically detect membrane curvature, and motor proteins, or molecular motors. And so, even though we found a new pathway, we were able to publish this new pathway, we were still puzzled by how it works, especially since it uses so few components and it happens so fast. There must be a really robust physics-based mechanism for it. And indeed, the way to study this process, I don't really have time to get into the details of the experimental protocol, but we were building synthetic systems. So again, cells are way too complicated, way too many components that interact with one another. And so we tried to find the minimal components for this system. So as a model of a cell membrane, we reconstructed a synthetic cell membrane by using, by basically swelling lipid molecules into these spherical vesicles. Now we need to create a membrane tubule, and the way to do that, we made use of an optical tweezer. And this is a highly focused laser beam, which basically creates a gradient that then traps molecular objects or microscopic objects in here. And so we use this optical tweezer to deform the membrane and pull out this membrane tube. So here in the red channel, you have the membrane, and in the green channel, you have these proteins. So we did add these curvature proteins on the system. And then we observed, also having optical tweezers, we can then measure forces and be very quantitative about this process. Now, what we found that was really surprising is that we require mechanical force, a mechanical pulling force to actually finish endocytosis and break this membrane. So you can see another way to break this membrane is by actually reconstituting this whole system, but instead of us pulling on the membrane, we actually put in motor proteins in this system. So you'll see it in the movie here. So we put motor proteins in the system. They're walking along microtubules in a dish. They're pulling on the membrane, and they're breaking the tube. And this actually then ends endocytosis. So you still don't quite understand how this happens. So we decided to, and this is really difficult for us to kind of communicate. So to help communicate how what the underlying mechanism is, we drew up a little cartoon that was published on the cover of Cell, and basically what it really tells you what's happening here. So you have a cargo binding on the membrane. You have superhero here, which represents a molecular motor dynein. It walks along microtubules. She pulls on the membrane and forms membrane tube. These green things are membrane proteins that then bind on the membrane tube, stabilize the tube. But what they also do, and this is the key thing that we found, they form a friction barrier between the tube and the underlying membrane. What this means is they prevent the equilibration of lipids underneath this protein scaffold and the mother membrane behind. And so as you are continuing to pull, as you are continuing to exert mechanical force, you keep destabilizing the tubules, you create micropores that finally destabilize the tube completely and initiate breakage and basically endocytosis. And so as physicists, we need to test our hypothesis. So what we did is we created various genetic mutations of our protein. And so we made a stronger binder to the membrane, a weaker binder to the membrane, and we can quantitatively see here that if you make a stronger binder, so this is supposed to say green is the stronger binder, orange is the weaker binder, it is based, and then we could see quantitatively that it fits our model, and then it confirms our hypothesis. Actually, I like this. That's another thing that we did to kind of explain this even further. And you can imagine a macroscopic analogy in which you're dipping a cooked noodle inside honey, where cooked noodle is a basically stabilized tube. And if you pull a cooked noodle out of honey, you're going to break it. If you're pulling it out of water, you're not going to break it, right? And the reason why it breaks is because there is friction due to the high viscosity of this of honey that prevents that destabilizes the tube. Okay, that's the mechanism. Now, what happens after cells receive this signal? They need to, at the embryo level, they need to interpret this signal so to pattern the tissue. And this is part 2 of this story. And I'm particularly interested in the symmetry breaking process, which happens at 2 weeks after fertilization. So we have a ball of cells of unidentified, morphologically, these cells don't look any different. Suddenly something happens at 2 weeks of development that separates the head from the posterior. This is called gastrulation. Due to ethical reasons, we're not quite allowed to study this in the actual human embryo. So instead, we need to find a proxy for the human embryo. And so over the past 2.5 years, I've been developing a synthetic system as a proxy for a human embryo. And essentially what it involves is using human embryonic stem cells, which I then grow in a 3-dimensional polymeric matrix. And then this polymeric matrix allows stem cells to self-organize into these beautiful epithelial shells, which, as you can see on the right side here, they're indistinguishable from a human 10-day-old embryo. So this is the 10-day-old embryo, and this is our synthetic embryo. Not just based on these are transcription factors, and not just based on these expression of transcription factors, but also based on morphology, cell density, and other features. Okay, now what we do with this system? Now we need to provide it with those signals, right? These signals that need to enter cells. And so we worked hard to figure out which particular signals are required here to actually initiate symmetry breaking. But we did find that there is a particular early embryonic signal that we uniformly apply to our synthetic embryo and does indeed differentiate these cells into something that looks a pre-head versus a pre-bottom. So I'm showing you a movie here. These are CRISPR-Cas9 mediated cell lines which have knocked in fluorescent reporters for various different cell fates. In green, you will have an anterior fate, so representing the head, and in blue, you will have a posterior fate. And so what you'll see here, that during the course, just after I add a signaling protein called BMP, something like 30 hours later, you will have the emergence of the posterior cells on one side of the synthetic embryo, and the anterior cells being upregulated on the opposite side of this synthetic embryo. And I have to say, this is the first ever live observation of a symmetry breaking in human. So how do we even go about understanding this mechanism? Go back to CRISPR-Cas9, the beautiful gene editing technology, and now we make informed decisions which genes to knock out. So we design a list of genes, we systematically knock them out one by one, and then we repeated this experiment to see how it affects symmetry breaking. And eventually we found one particular gene called DKK. This is an inhibitor of early differentiation pathways, and we found that once we knock it out, we vastly change the way the system is patterned. So here you have the knockout. As you can see, it uniformly forms the posterior, so basically it's all but, compared to the wild type, which is asymmetric, posterior on one side and anterior on the other side. Good. So now how do we, what's really going on, right? Well, we are adding a uniform signal, but somehow we found which signals are responsible for this, but how do we go from anterior, how do we get one side of the embryo to pattern one way and the other side of the embryo to pattern the other way? Well, again, another mathematician solved this problem. And this happened something like maybe 70 years ago, that Alan Turing was also very fascinated by biology. He believed that the spots and stripes on animals are a consequence of diffusion reaction mechanisms. And so he basically, by applying his model, we realized that if we apply a feedback loop of an activator and inhibitor, we can turn, especially using our DKK, we can turn our uniform signal into something that is asymmetrically patterned. Okay, and so what I've been telling you today, to summarize, even though these two projects seem very different, but they actually have a common theme, and this is understanding how we start from a signal that enters the cells. We describe the new endocytosis pathway, and then how these signals are interpreted to form a pattern tissue. And so what I intend to do in the future, hopefully in my own lab, very, 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 very soon, is now build on these synthetic systems, build on these synthetic embryos, and this is what I'm showing you here is actually one of my most recent breakthroughs, is where I was able to reconstruct an entire human embryo. It's embryonic and extra-embryonic tissue, so things that will become placenta. And I hope that these experimental models, coupled with mathematical modeling, are going to be able to answer important questions, such as fundamental questions, such as signaling at times when we're not allowed to look at it in the real embryo, perhaps help in bioengineering organoids and organs on chip in a much more quantitative way. And I'm sure that it's going to stir up some interesting bioethics debates, which is a very healthy thing in science. All right, and finally, I was told that I should present to you my trajectory, how I got here. I should probably start where I was born, and that's in a country that doesn't exist anymore. Right now, it's called Croatia, hopefully for a long time. I spent the majority of my early childhood in the middle of a war zone, and I did manage to get out of that. And really I'm trying to tell you is we don't really choose where we're born, but if you're really, really, really lucky, you can choose to pursue your passions. And I was the one who was really, really, really lucky. So I was able to pursue my passion for science, first starting at the University of Chicago, where I studied theory, and then later at the Institut Curie in Paris, where I studied experimental physics. And finally now I'm a member of Simon Society of Fellows. I'm a junior fellow and also at the Rockefeller University, where I'm super lucky to be studying these very exciting problems. Thank you for your attention.